Welcome back to Clinical Hematology Reviews. Today, we'll be discussing hemoglobin. Hemoglobin is really the cornerstone molecule of red blood cell function, and understanding its quantitative indices allows us to immediately evaluate cellular morphology and diagnostic anemias in the clinical laboratory.
Absolutely. When you step back and look at the erythrocyte as a whole, it is essentially a highly specialized vessel optimized for gas exchange. Hemoglobin is the primary intracellular protein driving that process, taking oxygen from the lungs to oxygen-starved peripheral tissues and helping return carbon dioxide back to the lungs for exhalation. What is fascinating is just how dense it is within the cell. Hemoglobin makes up roughly 33% of the total red blood cell volume, and if you dry out an erythrocyte completely, hemoglobin accounts for about 90% of its dry weight.
That density is precisely what gives red blood cells their oxygen-carrying capacity. To measure and evaluate hemoglobin content in routine patient samples, we rely heavily on specific quantitative indices from a standard automated complete blood count. First, we have total hemoglobin concentration, which normally spans from 12 to 17 grams per deciliter in healthy adults, though reference intervals shift slightly based on sex and age. Then, to look at individual cells, we evaluate the mean corpuscular hemoglobin, or MCH, which measures the absolute weight of hemoglobin per red cell, usually sitting between 28 and 34 picograms.
And we cannot forget the mean corpuscular hemoglobin concentration, or MCHC, which evaluates the concentration of hemoglobin relative to the actual volume of the red blood cell. Normal MCHC ranges from 32 to 36 grams per deciliter. In the clinical lab, MCHC is our primary indicator of cellular chromicity. A low MCHC signals hypochromia, meaning central pallor exceeds 1/3 of the cell's diameter, whereas an elevated MCHC often points to spherocytosis or significant cellular dehydration.
Now, to understand how hemoglobin carries out its physiological job, we have to look closely at its quaternary protein structure. A mature, functional adult hemoglobin molecule is a tetramer, meaning it is constructed from 4 globular protein subunits called globin chains, along with 4 embedded heme groups.
That tetrameric design is brilliant. The 4 globin chains are arranged as 2 identical alpha-like chains and 2 identical non-alpha chains. The alpha-like gene cluster produces alpha and zeta chains, while the non-alpha gene cluster produces epsilon, beta, delta, and gamma chains. In normal adult circulation, the classic combination consists of 2 alpha chains paired with 2 beta chains. Nestled inside a hydrophobic pocket of each globin chain is a heme subunit, an iron-chelated protoporphyrin IX ring.
That hydrophobic pocket is critical because it shields the ferrous iron atom from being spontaneously oxidized by water in the plasma. Because each of the 4 heme rings contains 1 central ferrous iron atom, and each iron atom can reversibly bind 1 molecule of oxygen, a single tetrameric hemoglobin molecule can transport up to 4 oxygen molecules simultaneously. To appreciate how these complex tetramers are built, we need to trace heme synthesis, which is an intricate assembly process spanning 2 compartments, the mitochondria and the cytoplasm.
That compartmentalization is key to regulating the pathway. Heme production actually begins inside the mitochondria, where the enzyme delta-aminolevulinic acid synthase condenses glycine and succinyl-CoA to form delta-aminolevulinic acid, or delta-ALA. This initial reaction is the master rate-limiting step of the entire pathway, and it is strictly regulated by the concentration of intracellular ferrous iron. If iron levels are deficient, delta-ALA production halts right at the starting line.
Once delta-ALA is formed, it exits the mitochondria into the cytoplasm. A cascade of enzymatic reactions converts delta-ALA through a series of intermediate porphyrinogens, culminating in the production of coproporphyrin 3. Coproporphyrin 3 then travels back across the mitochondrial membrane into the matrix, where it is converted into protoporphyrin IX.
In the final step inside the mitochondria, the enzyme ferrochelatase inserts a ferrous iron atom directly into the center of protoporphyrin IX, yielding a complete heme molecule.
Once constructed, heme exits the mitochondria one last time into the cytoplasm, where it promptly binds to a newly synthesized globin chain.
While heme is being assembled across the mitochondrial membrane, globin chains are being actively translated on polyribosomes in the cytoplasm of nucleated erythroid precursors and reticulocytes.
The genetic arrangement governing this translation is very distinct. The genes encoding the alpha-like globin chains, alpha and zeta, are clustered on chromosome 16. Meanwhile, the non-alpha genes, epsilon, gamma, delta, and beta, are housed on chromosome 11. During early embryonic development, expression is restricted to zeta and epsilon chains. However, as hematopoiesis shifts from the yolk sac to the liver and bone marrow, alpha and beta chain synthesis takes over, forming the predominant globin backbone required for adult life.
As these globin peptide chains are released from the cytoplasmic polyribosomes, newly produced heme molecules slip into their designated hydrophobic pockets. The individual globin monomers rapidly pair up to form stable alpha-beta dimers, and then two dimers spontaneously assemble into the complete four-part tetrameric hemoglobin structure. What amazes me about erythropoiesis is the extraordinary balance required. Under healthy conditions, the cell produces alpha chains, non-alpha chains, and heme molecules in an exact 1:1:1 ratio.
That balance is essential. Any significant mismatch leads to free chain precipitation, membrane damage, and reduced cell survival, which is precisely what occurs in thalassaemic disorders. The primary regulatory checkpoints include iron concentration, the availability of functional heme biosynthetic enzymes, and direct translational feedback.
That translational feedback mechanism is particularly elegant. Inside the reticulocyte cytoplasm, there is a specific mRNA translation inhibitor. When intracellular heme concentration rises, heme directly binds and inactivates this inhibitor. This allows ribosomal translation of globin mRNA to proceed smoothly. Conversely, if heme levels drop due to iron deficiency, the inhibitor remains active, shutting down globin synthesis so the cell doesn't waste energy accumulating free, unstable globin chains. As an individual matures from intrauterine life to adulthood, the specific hemoglobin tetramers synthesized change dramatically in a tightly controlled developmental sequence.
In the first trimester of embryonic gestation, we observe embryonic hemoglobin synthesized in the yolk sac, specifically Gower-1, composed of 2 zeta and 2 epsilon chains, Gower-2, made of 2 alpha and 2 epsilon chains, and Portland, consisting of 2 zeta and 2 gamma chains. By the end of the first trimester, embryonic hemoglobins disappear completely as fetal hemoglobin, or HbF, takes over. Hemoglobin F is composed of 2 alpha and 2 gamma chains, and it dominates throughout the remainder of fetal development.
Hemoglobin F has a remarkably high oxygen affinity, which enables the fetus to pull oxygen directly from maternal circulation across the placenta. Shortly before birth, beta chain production ramps up significantly, driving a transition to adult hemoglobin types. By 6 months of age, healthy individuals establish their permanent adult hemoglobin distribution, greater than 95% hemoglobin A, composed of 2 alpha and 2 beta chains, roughly 1.5 to 3.5% hemoglobin A2, composed of 2 alpha and 2 delta chains, and less than 2% remaining hemoglobin F.
It is also worth pointing out that hemoglobin A includes a glycated subgroup called hemoglobin A1C, normally making up roughly 3.5 to 5.5% of total hemoglobin. Because glucose binds non-enzymatically to the beta chain N-terminus over the cell's 120-day lifespan, hemoglobin A1C provides an invaluable clinical window into long-term glycemic control in patients with diabetes.
Now that we understand the structure and developmental expression of hemoglobin, let's explore its core function, oxygen binding and transport, represented visually by the oxygen dissociation curve.
Hemoglobin exists in 2 distinct structural states: the deoxy or tense state, the T state, which has low oxygen affinity due to inter-subunit salt bridges, and the oxy or relaxed state, the R state, where oxygen binding breaks those bridges, causing the molecule to shift into a high affinity conformation. Because the binding of one oxygen molecule increases the affinity of the remaining subunits, hemoglobin exhibits cooperative binding, resulting in a classic sigmoidal or S-shaped dissociation curve.
A fundamental metric on this curve is the P50 value, defined as the partial pressure of oxygen at which 50% of hemoglobin is saturated. In normal human adult blood, P50 occurs at approximately 26 torr, or 26 millimeters of mercury.
If the curve shifts to the right, the P50 increases, meaning hemoglobin has a lower affinity for oxygen and releases it more readily to hypoxic, metabolically active tissues.
Exactly. Right shifts are triggered by conditions typical of exercising muscle or ischemic tissue, elevated partial pressure of carbon dioxide, decreased pH or acidosis, also known as the Bohr effect, increased temperature, and elevated levels of 2,3-bisphosphoglycerate. Conversely, a left shift indicates a decreased P50 and higher oxygen affinity, meaning hemoglobin holds onto oxygen tightly. Left shifts occur in pulmonary capillaries where temperature and carbon dioxide levels drop, pH rises, or when high-affinity hemoglobins like hemoglobin F are present.
Hemoglobin's ability to alter its oxygen affinity in response to chemical signals relies on its allosteric properties, most notably its interaction with 2,3-BPG, also known as 2,3-diphosphoglycerate or 2,3-DPG.
2,3-BPG is an organic phosphate produced within erythrocytes via the Rappaport-Lubering glycolytic shunt. It acts as a critical negative allosteric effector. In tissue capillaries where oxygen levels drop, one molecule of 2,3-BPG binds directly into the central cavity of deoxyhemoglobin in its tense state in a strict 1-to-1 ratio. This cross-links the beta chains, stabilizing the low-affinity T state and forcing the release of bound oxygen into the tissue microenvironment.
Then, when those red blood cells travel back to the high oxygen environment of the lungs, high partial pressures of oxygen force oxygen onto the heme iron. As the third oxygen subunit binds, the entire tetramer snaps back into the relaxed state. That structural shift physically constricts the central cavity, expelling the 2,3-BPG molecule and preparing hemoglobin to load its full payload of oxygen once again.
Oxygen unloading at the tissue level is directly paired with carbon dioxide transport back to the lungs. Tissues produce carbon dioxide constantly through aerobic metabolism, and the red cell handles its return trip via 3 distinct mechanism pathways.
The majority, roughly 70% of tissue-derived carbon dioxide, diffuses into the red cell cytoplasm and is rapidly converted by carbonic anhydrase into carbonic acid. Carbonic acid spontaneously dissociates into hydrogen ions and bicarbonate. Bicarbonate then diffuses out of the red cell into the plasma, while chloride ions move inward to maintain electrical neutrality, a phenomenon we call the chloride shift.
The second pathway accounts for roughly 23% of carbon dioxide, which binds directly to the uncharged amino terminal groups of globin chains in deoxyhemoglobin, forming carbaminohemoglobin. Because deoxyhemoglobin binds carbon dioxide with higher affinity than oxyhemoglobin, oxygen delivery at the tissue level directly enhances carbon dioxide loading. Finally, the remaining 7% of carbon dioxide simply dissolves directly into the plasma as free gas.
At the end of an erythrocyte's normal 120-day lifespan, senescent cells must be removed from circulation and their molecular components systematically recycled. Under normal physiological conditions, roughly 90% of red cell destruction occurs via the extravascular pathway.
Extravascular destruction takes place when aging, non-deformable red cells are phagocytized by macrophages of the reticuloendothelial system, situated primarily in the spleen, liver, and bone marrow. Inside macrophage phagolysosomes, the hemoglobin molecule is taken apart with remarkable efficiency. Globin chains are cleaved by proteolysis into free amino acids, which return to the systemic pool for new protein synthesis.
Meanwhile, the heme ring is opened by heme oxygenase. This cleavage liberates carbon monoxide, which travels in blood to the lungs to be exhaled, and yields biliverdin. Biliverdin is rapidly reduced to unconjugated bilirubin, bound to plasma albumin, and transported to the liver for conjugation and biliary excretion. Iron liberated from heme is either stored locally inside macrophages as ferritin and hemosiderin, or exported onto plasma transferrin, which carries roughly 80% of our daily functional iron pool directly back to the bone marrow for fresh erythropoiesis.
While extravascular phagocytosis handles 90% of normal red cell clearance, the remaining 10% of RBC breakdown occurs directly within circulating blood vessels, a process known as intravascular destruction.
When a red cell lyses directly inside a blood vessel, free tetrameric hemoglobin is released into the plasma and rapidly dissociates into alpha-beta dimers. To prevent free hemoglobin from causing oxidative tissue damage or renal toxicity, the plasma transport protein haptoglobin immediately binds these free dimers in a high-affinity complex. The haptoglobin-hemoglobin complex is far too large to pass through the renal glomerulus, so it travels safely to the liver, where hepatocytes clear it from circulation.
However, during severe acute hemolytic crises, intravascular lysis surges, quickly saturating and completely depleting plasma haptoglobin levels. When uncomplexed alpha-beta dimers remain in plasma, they are small enough to pass through the glomerular filtration barrier into the renal tubules.
Proximal tubular epithelial cells attempt to reabsorb these filtered dimers and convert the iron into storage proteins. But when renal reabsorptive capacity is overwhelmed, sloughed tubular cells containing iron pass into the voided urine. Detecting this iron precipitate via Prussian blue staining confirms hemosiderin, a classic diagnostic hallmark of significant ongoing intravascular hemolysis.
To conclude our review, we must examine acquired non-functional hemoglobins. These are altered forms of hemoglobin that lose their ability to reversibly bind and transport oxygen, presenting clinically with tissue hypoxia, cyanosis, and altered blood coloration.
The first variant is methemoglobin, which occurs when the iron atom within the heme ring is oxidized from its functional ferrous state to the ferric state. Ferric iron cannot bind oxygen. While small amounts of methemoglobin are continually generated, the methemoglobin reductase pathway normally keeps levels negligible. When chemical exposure or oxidative drugs overcome this enzyme system, methemoglobin accumulates, turning the patient's blood a distinctive chocolate brown color and causing cyanosis that fails to improve with supplemental oxygen.
The second variant is sulfhemoglobin, which develops when a sulfur atom is permanently incorporated into the protoporphyrin ring, often following exposure to sulfonamides or sulfur-containing medications. Sulfhemoglobin cannot bind oxygen at all, and because the chemical alteration is completely irreversible, the affected hemoglobin remains dysfunctional for the entire remaining lifespan of the erythrocyte.
Finally, we have carboxyhemoglobin, formed when carbon monoxide binds to the heme iron. Hemoglobin has an affinity for carbon monoxide that is roughly 200 times higher than its affinity for oxygen. Carbon monoxide binding locks the remaining subunits into an extreme high-affinity relaxed state, shifting the oxygen dissociation curve far to the left and severely impairing oxygen unloading to tissues. Clinically, patients present with severe hypoxia despite displaying classic cherry red discoloration of the blood and skin.
That comprehensive breakdown covers everything from synthesis and structure to function, degradation, and pathology. Excellent review today. Thanks for joining us.